The overall goal of this protocol is to study the direct and indirect communication between primary breast cancer cells and monocytes in a three-dimensional co-culture system.
This method can help answer key questions in the inflammatory microenvironment of breast cancer field, such as how immune cells are recruited and activated, how these cells help to sustain the tumor inflammatory microenvironment, and how this microenvironment facilitates tumor cell invasion.
The main advantage of this technique is that it provides an affordable alternative to study intratumor communication and illustrates the potential of in vitro 3D cell systems for interrogating specific features of tumor biology, particularly those related to tumor aggression.
To begin, cultivate 2 × 10⁶ U937 and THP1 monocytes in 10 milliliters of rPMI 1640 medium, supplemented with 10% FBS and 1% antibiotic antimycotic. Also cultivate fresh PMs in supplemented DMEM F12 medium. Incubate the cells at 37 degrees Celsius in a humidified 5% carbon dioxide environment. Plate 4 × 10⁵ monocytes in 1 milliliter per well of their corresponding medium. Place an insert in each well and add 900 microliters of the 4 × 10⁵ BRC cell suspension in the corresponding medium. Incubate the cultures at 37 degrees Celsius in a humidified 5% carbon dioxide environment for 5 days and recover the supernatants.